Thank you. All right. So so today I guess I want to go through and detail some new approaches in spatial population genomics that we've been applying to this particular invasion. And this idea of recent invasions is actually quite interesting here because they're very interesting systems. Recent invasions are, you've got some species has just showed up somewhere new, and there's often great. It's very dynamic. There's a lot of change happening very very quickly, but they're also then quite difficult to actually analyze with standard. A lot of traditional population genetics approaches are thinking about populations at equilibrium and moving around. Recent invasions just throw all of that out the window, and it becomes really, really complex to analyze them. So we need to think about new ways in which to investigate them. And that's also very important because these recent invasions can be very, very important. This one in particular is extremely important. I mean, this is an invasion of Aedes albopictus, aka the tiger mosquito, aka the Asian tiger mosquito, aka the barbecue stopper. And it's called the barbecue stopper because wherever it's gone in the world, it's out during the day, it's biting constantly, and it spreads diseases like dengue, Zika, chikungunya, etc. So this mosquito is basically found, it's from East and Southeast Asia. It's spread through Europe. It's spreading north through Europe. I think it's reached Switzerland at this point. It's spreading west through the United States. And it's reached Australia technically because it has reached the Torres Strait Islands. And for those who don't know, the Torres Strait Islands is this area up here, which is in between Cape York, Australian mainland, and this area in southern PNG called the Southern Fly region. And the Torres Strait Islands, so here's an example of one of the islands. This is Masig Island. This was where the mosquito was first found in 2004. And you can see this is not some resort island. This is not some hotels or whatever. These are small villages. A few hundred people. But there's a lot of movement within the island. So people move by boat, people move by plane between the different islands, between the islands and the Australian mainland, and between the islands to and from Papua New Guinea. And it's actually human movement that spreads these mosquitoes. So the mosquitoes are really good at hitching a ride on boats, particularly, also on planes, on trucks, wherever. And that's what's happened here. Basically, the mosquito. So these mosquitoes that were found, they spread into Papua New Guinea, but it seems chasing their genetics. Initial work on this was showing that the mosquitoes have an Indonesian genetic background. So they came from somewhere in Indonesia, and the invasion sort of spread into this. Let me get my laser point. Into this area, the Southern Fly region, and then from there into the Torres Strait. So we don't know exactly how it spread, the exact dynamics of whether it spread to one island first and then through there. But what we do know is that basically before 2004, this mosquito effectively wasn't in the Torres Strait, and then by 2006 it was everywhere. So it had just rapidly spread through the whole system. So the idea, this is currently the situation we're dealing with. People went in there, tried to wipe the mosquitoes out with insecticides. That didn't really work. So people set up this what's called a cordon sanitaire here, which is basically a barrier saying if you want to travel from the north past the cordon sanitaire, there's going to be extreme restrictions on the travel. So we're going to try and stop the mosquitoes spreading further from here. But we still keep finding mosquitoes down in these islands here. So there's basically, and this is very close to the Australian mainland. So the idea is we're going into this project. The idea is we want to work out dispersal in this system in a really a thorough way. We really want to understand how the mosquito is moving around in this greater area, Papua New Guinea, Torres Strait, through the Torres Strait, and then also invading into new areas. And there's a really big push to stop this thing from reaching the mainland. Now, often with looking at, you can look at a set of spatial genetics. You can look at a sample across an area and look at some genetic structure. Now, this is what it looks like here, for instance. This is, some of our findings, and this is a simple admixture plot showing two genetic backgrounds, the blue and the gray background. And we can see, there's a bit of spatial structure here, and there's also some individuals you see, say here, there's the one individual with a blue background, and it's all the other individuals on this island have got a gray background mostly. The thing is, because this is a recent invasion, we can't just do this and then infer that there's been all this movement going on. We have to treat the problem quite differently because remember, this is a recent invasion. The whole system has a lot of recent co-ancestry. So we're looking at changes that are happening here, from generation to generation. This is not some very, very deep time study. We're looking what's happened very recently. We expect everything to be similar, but we need to somehow detect within that general region of broad similarity. So in talking about dispersal, there's really, we want to try and characterize this thoroughly, but throughout the 20th century, if you and to now, really, if you're asking biologists what is dispersal, then depending on who you ask, you might get a different. From a population genetics perspective, dispersal is an intergenerational process. So you're looking at something where the individual's movement is made up of individual movement, but the dispersal itself is sort of abstracted beyond that to a process. So here we have what's called a spatial pedigree. So this is a genealogy we've got time here, genealogy of all these individuals, which are circles collected at a given time point, but it's also connecting them through space. So we have, say, these two individuals, one generation back, are connected. So these individuals will be siblings, and this is the parental generation. If you go back another generation, that'll be the grandparental generation. So if you share a pair of grandparents with someone, then you're cousins. So it's either one generation siblings, two generations cousins. So you can look here with this idea of kinship, and that just sort of plays out, generally speaking, over as much time as you want to create this spatial pedigree. But then on the other hand, you've got this situation of if you talk to an ecologist, say, they might say, well, dispersal is individual movement. And that's not, it doesn't have to have an intergenerational effect, because sometimes it's important that individuals move around. So I've put a picture of plankton here, for instance. So plankton, might be a lot of little larvae that are going along, bobbing along happily in the plankton while they're developing, and then they move somewhere, they get eaten without reproducing. So they have not contributed anything to the next generation, but that movement is very important from a food web perspective. So that movement is still important. And the idea here is clearly movement across generations, movement within generations. What I'm getting at, and what I want to get across here, is this idea we're now, with population genomic approaches, we can actually start not looking at either or, we can look at both of these at the same time. So here's some examples, recent examples from mosquito work. And what we have is, we have this, so we've got two plots here basically on they're both lines connecting up dots, but they're describing different things. So on the left here, what we have is this intergenerational dispersal idea. So these lines connect up full siblings. These are half siblings, and these are cousins. They're polygons. This is an area in Kuala Lumpur, Malaysia. And these are apartment buildings, and we're seeing the distribution of, so these individuals are one generation separation, these are two generations separation, and we're looking how far apart they are. So that's intergenerational movement. On the other hand here, this is actually movement within a generation. So we have in white, this is Aedes albopictus mosquitoes. In red, these are Aedes aegypti. And what's happening here is we're collecting these mosquitoes as they disembark from an aeroplane or from a boat. So they're being collected at Australian biosecurity at ports, airports, seaports, and we're using genomics to work out where they came from this panel of reference population. So you can see here, it's quite interesting. These mosquitoes are actually coming from different locations. So we have movement across generations. We've got movement within it generations. This is using population genomics, but it's using population genomics in this loose kind of broad sense here, where it's really it's taking what we were doing before and just adding more markers to it. So you have one marker, you have one SNP. Is that population genomics? No, but if you go to 10, oh, not really. No, once you get to 10,000, okay, now it's population genomics. It differs quantitatively, but it might still be asking the same sorts of questions. So this is generally called broad sense, and then you've got narrow sense population genomics, or true genomics, where you're using this general genome-wide context to find particular areas of interest. Now, there's something to say, okay, this is real genomics and this isn't genomics. I think that there is a lot to recommend that point of view. There's a lot to recommend that argument. However, I do think it's worth paying attention to the fact that when you have a lot of markers, you can ask a whole different set of questions. So I'll give you an example here just from before. With microsatellites, you can detect full siblings, maybe half siblings, but not cousins. And now we can actually get all of them. And bear in mind, this is two generations separation. This is one. And my colleague Mosh Jasper actually showed that when you have 2 generations of data as opposed to 1 generation of data, you can actually then use that to parameterize dispersal in the population in a way that you just couldn't do otherwise. So while this is not genomics in a narrow sense, there is something different going on there. And there's a quote variously attributed to Stalin, Lenin, and Napoleon, which is, quantity has a quality all of its own. Now, they were talking about infantry, but I think you can also use it to refer to SNPs, because once you have enough of them, you can categorically change the sorts of investigation that you do. But so that's, looking at broad sense. Narrow sense genomics, you can also look at dispersal, and we also did, using this, investigating in this way. And for invasive species, one of the ways which this is really useful is looking at genetic invasions. So genetic invasion is when you've got an invasion happen. So the mosquito has arrived, and then you get some more genetic material coming from somewhere else, and it makes the invasion harder to control. So a classic example with that would be with adaptive introgression, and that's an example here. This is with an Aedes aegypti mosquito from. We're looking across the Indo-Pacific regions. If you look at the 3 chromosomes, generally speaking, they're just the genetic structure is purely spatial. But once you look within the voltage-sensitive sodium channel gene, it's really not. And within this gene, there are 2 mutations. If you're a homozygote for one mutation or the other mutation, you go from being susceptible to pyrethroid insecticides to being really resistant to them. So it's really useful to have it if you're a mosquito and there's a lot of spraying insecticides going on. So it seems here, from when we looked at this, it's likely that these 2 mutations only evolved once each in the Indo-Pacific region, and then just spread through the whole region via genetic invasion. So here we have, we've got this recent invasion. Now, if we approach it somewhat nihilistically from a population genetics point of view, we'd say, all right, well, I'm not going to trust any of these the standard assumptions of equilibrium. We're not going to assume that the things had time to settle down. What can we trust? And here's what going through, I would argue that these bits of information we can place some trust in. We can actually understand what's going on. So one thing is finding individuals from outside of the invasion and working out where they came from. Another one is actually identifying pairs of kin from within the invasion. So you have this kind of morass of allelic variation, and you're just pulling out pairs of kin from that you say, okay, well, you two are closely related. There must be a recent dispersal event going on there. And then there's also adaptive introgression. If we can't tell much, but if the whole invasion is all very similar, we can at least tell if genetic information has come from outside into it. So here's the system, and here's basically how sampling takes place. If you want to do this sort of study, sampling is really key. It's generating sequence data is fine, but you've got to get the sampling right. And here's the Queensland Health went and did this sampling. They did a fantastic job. So here's Masig Island again. Here's what sampling looked like on Masig Island. Here you have so all of these yellow circles is where a Queensland Health worker stood and, using themselves as bait, trapped Aedes mosquitoes that came to bite them, right? And then doing this, but throughout the entire Torres Strait region within a 2-week period. So that's really 2 weeks, that's a generation of mosquitoes. So we collected throughout the whole area within about 1 generation. And then, as well as that, we had mosquitoes collected below the cordon sanitaire. So they're invading, and we're sort of capturing them before they've established an invasion, and then working out where they came from. And then finally, we're looking at movement into the Torres Strait from this broader panel of Indo-Pacific genotypes. Sort of say about the sequencing approach we're using here. So this is with double digest RAD sequencing, which is a type of reduced representation sequencing. It sequences about 1% of the genome, and there are some downsides to reduced representation sequencing. There are some upsides to it. I mean, the downside is you're only getting a bit of the genome, but if you have a very good reference assembly, and thankfully there's just been released just in the nick of time for us, a very, very good reference assembly was put out for Aedes albopictus. If you have a good reference assembly, you can get a lot out of it. And of course, when you're only sequencing a little bit from each individual, comparatively, you can get a lot more individuals, and that actually changes what sort of questions you can ask. So a comparison here, as well to look at is Anopheles gambii, which is the major malaria vector in Africa. So this species has a very small genome compared to Aedes albopictus. It's a fifth of the size. So this was actually sequenced around 2002. So it was really easy to sort out this genome assembly. And most genomic work on Anopheles gambii is investigating with whole genome resequencing. So there's about 1,600 whole genomes up at the moment, and that number is just being added to all the time. For Aedes albopictus, that would cost 5 times as much for the cost of sequencing, and you also have a huge genome. 1% is still quite a lot. So here's what we found overall. So tracing these incursions, now we use this method that's just come out called Locator, and Locator is really good. It's a deep learning method, but what's cool about it as well is that it treats genotypes individually. So it's basically saying, all right, we're looking at, we've got 4 incursive mosquitoes, 1, 2, 3, 4. Let's take this, this particular genotype and compare it to all these other genotypes as a spatial location. So it's not, say, aggregating, saying, all right, these individuals here are one population, this is a population, this is a population, because those, we're not going to assume that. We're not making a hypothesis that these are populations. We don't know. So it's better to just treat the genotypes in space as they are. And we actually found this outperformed another method that was using where you just use allele frequencies based on the population. So you can see here, what we have is all of these transparent red circles. There's 1,000 of them in each subplot, and they are showing these are bootstrap replicates of where it's placing the mosquito. So where is it likely from? So here it seems very likely from this one island nearby called Kariri. And then for this one, it seems to be placing it with a bit more variation, certainly, but seems to be placing it on this big island to the north called Moa Island. And then here, it just has no idea. We couldn't sort this individual out. So yeah, there are limitations to this, but the fact that it was able to assign this one while the other programs couldn't, show that this sort of methodology is actually really useful. We were also detecting kin. So that's where we sampled everything at the same time and then basically used, we were inferring relatives. We're trying to pick out relatives from their genetic information. And you can see on this plot, basically these shapes that are the same color show a pair of kin. The square with the black dot shows where the origin is, and then it's dispersed to here. So they're all basically between nearby islands, apart from this yellow one, which went all the way from here over to here. There's about 202 kilometers. And this is where having lots of individuals is cool. We've got a lot of information. We have enough genetic information to infer kin, but having a lot of individuals is very useful. So we had 301 Aedes albopictus we sequenced from here, but that actually gives you 45,000 pairwise comparisons. And then across different villages, it's around 40,000. So out of that 40,000, we found 5 individuals. So it's not a huge number, but bear in mind, we can be quite confident about this. We're not trying to assume a rate. We're not trying to, there are a lot fewer assumptions to just saying, right, these are close relatives and they're on different islands. Something's just happened to make those on different islands. And you can see down here, this is basically, we have these, the red points are close kin found within the same island that we use to calibrate the model. There are 3 of them, and then there are the 5 orange points that are the close kin dyads across different islands, and then there's just everything else. So there's quite a big window of daylight between the ones we've assigned as kin and the ones we're just saying are closely related individuals of indeterminate kinship. We can also actually look within islands here, which is pretty cool, because this is basically, we're not looking at passive movement anymore. We're not looking at movement of the individual on a boat or on a plane. We're looking at it flying around its own island. And the same thing here, we're looking at the spatial distribution of genetic variation. So we can see it differs quite a lot from island to island. And a lot of this might be due to the fact we have a very recent invasion. Maybe it's just because there's so much movement between the islands. There's not this sort of set isolation by distance pattern. But we can calculate neighborhood size here, which is a measure it seems to be quite similar across mosquitoes so far when we've looked at it. So while no matter where the mosquito population is, it seems the sort of spatial distribution of genetic variation seems to be pretty similar once you look at a fine enough scale. And then finally, what we found is a sign of a genetic invasion from Papua New Guinea. So this is basically we're looking for outlier SNPs that had strange structuring relative to the rest of the genomic context, and specifically SNPs that were in, like, where there are multiple SNPs together. So in a linked region, so we're seeing linkage. We're seeing where they were co-occurring, and then also where there were genes of known function that might be actually important. So we have this 135 megabase region, and then looking within there, there were 2 rad tags with outlier SNPs in very, very close linkage, and within this region there are 12 cytochrome P450 genes, and these are very strongly associated with metabolic resistance to insecticides. So note here, this is actually the comparison between the current genome that came out when we were first analyzing this, and we use this genome assembly, and this is the previous one. So we have the N50 of scaffolds is just tremendously different between these. So this is basically, if you take the top 50% of scaffolds in terms of length, the shortest one of them is this long, while the shortest one of them is this long. So if we were trying to do this sort of analysis with the previous genome, we may not have gotten anywhere with it. So even though you've got reduced representation, very, very sparse sequencing, you can actually get pretty good outcomes if you've got a really nice genome assembly, and it's definitely getting better for albopictus at the moment. But finding some resistance is not in and of itself that interesting. I mean, we know we try and kill this mosquito constantly all around the world. It's one of the few things we agree on, is that this mosquito sucks, right? And so we've been bombarding it with insecticides, and we expect to see something happening there. What's more interesting is where they're coming from, because when we looked at it, we found the alleles were all in the Torres Strait. They were all in southern Papua New Guinea, and then nowhere else. There was just one individual here in Singapore. I think that's just a red herring. But basically, no sign of any of this stuff in anywhere, in Indonesia, in Timor, but in southern Papua New Guinea. So it seems the invasion may have come in through Indonesia, but then been sort of fed some insecticide resistance alleles during the early stages of the invasion. Maybe when they were spraying a lot of insecticides at the start, that's when that happened. So yeah, I'd just like to finish up saying, this is sampling across, a huge area, and anyone who's worked in, with sequence data knows, you end up with this enormous amount of data. You have, collecting and getting sequence data is becoming easier and easier, but you can't go back in time and sample differently. You're stuck with what your sampling is. So it's the importance of getting sampling right. And again, we had fantastic sampling from Queensland Health, meant that we can get, this analysis, this is tens of meters, hundreds of meters, a kilometer, tens of kilometers, hundreds of kilometers, thousands of kilometers. And that kind of spatial scale and the resolution across those scales is very, very nice if you want to really comprehensively do a study of spatial population genomics. I just thank everyone, co-authors, funding, etc.
Great, thanks very much, Tom. That's amazing work, amazing what you can do with 1% of the genome, that's for sure. Does anyone have any question? We have one in the forum. What does neighborhood size mean? Can you please elaborate on how it's calculated?
Sure, okay. Maybe I'll just have a, yeah, so neighborhood size is a concept from, so this concept is, it's an old concept from Sewell Wright, and the idea, so we have this equation, 4 pi times, this is the variance in distance between parent and offspring at the same life stage. So say from egg to egg, you're an egg, you start somewhere, and then you go around, and then you lay an egg somewhere. The distance there, that is the variance in that, not the mean. The variance is sort of that's dispersal in a population genetic sense, and this is density, the effective density. So basically, when you have a number, now I wouldn't say that I wouldn't treat this as an actual number of mosquitoes, but it's useful as a relative measure. The idea is basically you've got your neighborhood will be how far do you move and how many individuals are around as a density. So if you're moving a really big distance and it's really dense inhabitation, then your neighborhood size will be very big. But if you don't move very far and or you're very sparsely distributed, then you'll have a smaller neighborhood size. So these, yeah, it's something which has been, I think people are returning to a bit more now, because we can actually, it's been something that has been, so this term was brought about and conceptualized really in 1946, but it's really recently that people can actually explore it using any real data. So it's nice now we can go and mine all of the classics and just and start actually doing some comparisons.
Great. And we have another question. Can the locator tool be used for population level analysis and individual level analysis?
Can the locator tool be used for both population? Locator works specifically on genotypes, but yes, I mean, you could do it on populations if you basically just said that if you take a population being 20 individuals you sample from a given location, then you could certainly, you could, you could certainly just say, all right, well, that's a population. But I mean, there are so we also used another program called a SignPop, which is a machine learning approach, again, but this is using this is aggregating at a population level. So that's where the population is a set of allele frequencies for that location. So there are some circumstances where that might be more useful. I think the population level stuff is fantastic if you're looking at a broad scale. And as you get to a finer scale, the spatially explicit stuff for each genotype has a location becomes a lot more important, because and that's also true for any sort of recently disturbed situation, or where you might have you sample, say, like a broad region of genetic similarity. If you just like constant sampling rather than sample at point A, point B, you just sample everywhere in between, then it becomes very useful. And again, it's this idea of we can now, we're spoilt now for sequencing. We have amazing sequencing options and everything like that. The key is then to get the original stuff right, which is just to collect the right thing and actually record where you found it.
Great. Another one popped up. Wonder if invasive pest control measures should be modified given the strong selective pressure for insecticide resistance that you showed.
So I can't see.
Sorry, just about whether pest control measures should actually be modified based on the examples like the insecticide resistance you showed. And
absolutely. Yeah, absolutely.
I guess in the future.
Yeah, so that's, I mean, that's something that, I mean, our group works very strongly on all this sort of stuff. So I mean, we're basically advocating for using endosymbionts, so such as Wolbachia, where you can actually sort of crash or control populations using beneficial bacteria rather than using insecticides. But if insecticides are to be used, one of the options is to just rotate them around so that you have if you've got a pressure from one type of insecticide, and then that selects for resistance, then switch to a different type of insecticide, and then and that will then sort of the population will have to keep up. But of course, one of the things we're seeing from here with genetic invasions is that you don't need to, if you switch to a new insecticide, it's not like the population needs to mutate all this stuff perfectly by itself. It can just have some new alleles come in very quickly. And that means you can have resistance spreading very rapidly.
So, all right, I don't know if you can hear me.
Yeah, I hear you.
Oh, you can. Good. Hey, really great stuff, Tom. I enjoyed that. And one thing I wonder, the idea of the resistance is they no longer control Aedes albopictus on those outer islands. So the outer islands are everything outside of that cordon sanitaire. And I'm wondering if you've noticed a potential loss of resistance because it's not being selected for. There obviously could be stuff coming in from PNG to feed into it, but you would think with time that those alleles might be selected out. Do you have any feeling about that and how long it might take, how many generations it might take?
Yeah, well, I mean, in terms of how long it might take, I'm not really sure. You'd be able to say something better about how much local, I mean, presumably if there's no sort of, the Queensland government or the Australian government is not doing any spraying there, maybe the people who live there might be doing it themselves if there's still some selection on it.
Yeah.
But it seems that what's happening, see, we found that genetic invasion, but it wasn't at a very high frequency. It was really just on, one island, Masig, and then on a couple of islands there was a little bit of it. But it does, if there was strong selective pressure now, we'd expect to see those alleles at a much higher frequency, I think, if indeed that's what
Yeah.
it's representing, and that would be spread. So it seems like it's sort of spread in there when this is my, I don't know, I can't back this up, but I think is, good explanation might be that this came in early on in the invasion when there was a lot of spraying. They finished, 2008,
Yeah.
2009, and then it's just been sort of stalling there at the moment.
Yep. Yep. Okay, thank you.
Great.